Today, we are going to discuss the role of the immune system in host-microbe interactions, which is a crucial topic in dairy technology. In this session, we will understand this topic step by step. Now, let us understand an introduction to host-microbe interactions. This involves various players. First, we have bacteria, which constantly interact with their environment. Then we have fungi. These microbes exist within a host environment, forming a complex microbial community. All these elements engage in a dynamic interaction which shapes the health of the host. Next, we will focus on the meaning of host-microbe interactions. These interactions can have different outcomes. They can be beneficial or neutral, meaning they do no harm. On the other hand, they can also be harmful or damaging to the host. Now, let us look at the two broad categories. First is coexistence. This includes mutualism, where both host and microbe benefit, and commensalism, where one benefits and the other is unaffected. The second category is conflict. This involves pathogenesis, which is the process of causing disease. So, in summary, we have symbiosis on one side and infection or conflict on the other. Another important part of this topic is understanding how microbes enter the host. They use major portals of entry. The first common route is the respiratory tract, where microbes are inhaled through droplets or aerosols. The second is through the skin or wounds via physical breaks, insect bites, or vectors. The third portal is the gastrointestinal tract through the ingestion of contaminated feed or water. And a very specific route in dairy animals is the teat canal, which is a primary site for assembling infection leading to conditions like mastitis. Now, let us see how the immune system recognizes these microbes. The first step is detection. Special molecules on the microbes called PAMPs bind to receptors on the host cells known as PRRs. This binding acts like triggering an immediate alarm. Following this, the antigen, which is a piece of the microbe, is picked up by an antigen-presenting cell or APC. This cell then presents the antigen on its surface using a structure called the MHC complex. This presented antigen is then shown to a helper T cell. This whole process of presentation is critical because it activates the body's specific defense mechanisms against that particular invader. With this idea clear, we can move further to the innate immune responses. These are the body's immediate non-specific defense mechanisms. The first line of defense consists of various barriers. We have physical barriers like the skin, mucous membranes that trap pathogens, and chemical barriers such as stomach acid or enzymes that destroy them. The next aspect involves cellular eaters known as phagocytes. These include cells like neutrophils, macrophages, and dendritic cells. Their job is to engulf and destroy invaders. Then we have the alarm signal, which is inflammation. When cells are damaged, they release chemicals called cytokines, which cause vasodilation or widening of blood vessels and recruit more immune cells to the site of infection. Finally, we have a soluble defense system called the complement system. This is a cascade of proteins that can coat microbes in a process called opsonization, making them easier for phagocytes to eat or even directly lyse or burst the microbe by forming a membrane attack complex or MAC. Now, let us understand the adaptive immune responses to microbes. This is a more specialized defense. It has two main arms. The first is the humoral response, which involves B cells. These B cells can mature into plasma cells that secrete antibodies. These antibodies travel in the blood and fluids to neutralize extracellular pathogens like bacteria by binding to them and preventing them from causing harm. The second arm is the cell-mediated response, which involves T cells. Specifically, cytotoxic T lymphocytes or CTLs directly seek out and kill cells in our own body that have been infected by viruses, thus stopping viral replication. A key feature of this adaptive system is immunological memory. After the first encounter, memory B and T cells are created. These cells persist in the body for a long time. If the same pathogen tries to infect again, these memory cells allow for a much faster and stronger response, which is the principle behind vaccination. Next, we will focus on the actual elimination of pathogens. The immune system uses several clever mechanisms. The first is opsonization. Here, antibodies or complement proteins coat the microbe, marking it for enhanced phagocytosis, making it easier for phagocytes to find and consume it. The second is neutralization. Antibodies can block a pathogen's attachment sites, preventing it from latching onto our cells, or they can also bind and neutralize harmful bacterial toxins. The third mechanism is direct lysis. The complement system we discussed earlier can form a membrane attack complex, creating pores in the microbe's membrane, causing it to take in water and burst from osmotic pressure. The fourth is phagocytic killing. Once a phagocyte engulfs a microbe, it encloses it in a phagolysosome where powerful enzymes and reactive oxygen species or ROS completely digest and destroy the invader. Another important concept is immune memory, which we can see by comparing primary and secondary immune responses. Let us look at antibody levels or titer over time. During the first exposure to a pathogen, the primary response is relatively slow and produces a lower amount of antibodies. However, if the same pathogen is encountered again, the secondary response, thanks to memory cells, is much faster, stronger, and produces antibodies for a longer duration. This is why we are often immune to diseases after getting them once or after getting vaccinated. Now, what is the ultimate goal of all these interactions? It is to achieve a balance or homeostasis between the host and its microbes. In a healthy state, the host maintains a beneficial microbiota, often called good bacteria. The immune system is in an avoid state, meaning it tolerates these beneficial microbes while keeping its barriers intact. When a threat appears, the system switches to an active state. It triggers inflammation and other responses to eliminate the threat. Once the threat is gone, the system returns to a balanced state. This dynamic equilibrium is crucial. Let us look at the possible outcomes of a host-microbe interaction after an infection. There are three main pathways. The first is clearance, where the immune system successfully and completely eliminates the pathogen from the body. The second is persistence. Here the microbe is not cleared but remains in the host, sometimes in a dormant or carrier state without causing immediate illness, a phenomenon known as latency. The third and undesirable outcome is disease. This is when the interaction leads to significant tissue damage and clinical symptoms of illness. Given all this, let us summarize the importance of the immune system. It plays several key roles. First, it maintains barrier integrity, ensuring our physical skin and chemical barriers like mucus are strong to prevent pathogen entry. Second, it enables vaccine response. Our adaptive immune system can be trained by our vaccines to develop memory and protection without us having to suffer from the actual disease. Third, it provides constant surveillance. The immune system continuously monitors all body tissues to detect any foreign invaders immediately. Finally, it is crucial for herd defense. The immunity of individual animals contributes to the overall protection of the entire herd, especially shielding those who are more vulnerable. To bring everything together, here's a visual summary of the complete pathway. It starts with the entry of the pathogen through portals like the skin, gut, or respiratory tract. Next is recognition, where host PRRs detect microbial PAMPs. This triggers the innate response involving inflammation and phagocytosis. Finally, if needed, the adaptive response kicks in with T cells and antibodies. This entire flow leads to the end result, which is either clearance, persistence, or disease. Thank you for watching. Stay tuned for more videos.